question that Val just asked about bush flies eating dung. I have another question to Fez. I noticed, Fez, in your talk, in your controls are around 25% dung removal without beetles and around 50% of dung removed with beetles, something like that. And I'm guessing that's due to earthworms, given that there are a lot of earthworms in New Zealand. And I'm wondering if you can partition effect of the worms from the beetles. And in experiments from the north of Europe, where beetles are rare, but worms are much more common, might give some insight.
Yes, I guess I didn't think about that or quantify earthworms. Although when I was taking soil cores, I did know that I did see some earthworms, but I'm not sure how I would factor that in to my results at the moment. But definitely New Zealand's sort of main strategy before 2011's approval of the dung beetles was to just chuck as many worms out there as possible. So yeah. I'm not sure if that answers the question.
You probably don't want to run another whole field experiment on this, but you might be able to run a smaller glasshouse type experiment where you add known quantities of worms as well as beetles.
Yeah, it's a really good question, actually. I think there's a researcher over in Tauranga, which is about 2 hours from here. There's just a scientific research institute, I can't remember what it's called, but she was interested in quantifying the earthworms effects on dung burial and dung beetle effects on dung burial. So there might be some data already out there that I can have a look at and link up with my research.
Yeah, New Zealand's a much wetter system than what many of us have here. So curious. And another question for you, and indeed everybody who's run any lab or field experiments, and they've also looked at nutrients. I'm wondering about timing of activity of the soil microbes and effect on various nutrients. So your samples, I couldn't remember when you do, and also depth of soil, because obviously most activity is happening in the top 10, 15, 20 centimeters of soil. So I wondered if you had not just different time periods, like time since the dung was available, but also the depth of soil. And if you saw any effect of either of those factors on the soil nutrients.
So I didn't split the soil cores to any depth value. There could be more of an interaction towards the center, I assume, of a 15 centimeter deep core, just simply because of the effects of evaporation and also just water. I guess maybe it's a Goldilocks zone. I just imagine it being like a more desirable zone for microbes, and those processes in that sort of middle area. I have no scientific backing to that statement other than that's what I sort of think that might be the case. But it would be interesting to see if there was any impact of depth, because I know that leachate does go a bit deeper, particularly in wet systems like New Zealand. And I think we have some collaborators looking at dung beetle effects on leachate, and I haven't heard what their results are at the moment, but there's definitely a potential of loss to deeper depths where they might still be contributing to a functional multifunctionality system, ecosystem effect. Yeah.
Yeah, interesting. Yeah. Have you got questions of any of the other speakers that are also doing functional ecosystem function experiments?
I have some notes. I was interested in Dan's talk. I think we're sort of trying to answer a similar question in terms of delivering multiple ecosystem functions. I was really interested in the evenness and density angle that he'd taken. And one of the graphs in one of his slides showed that as species richness increased, ecosystem functions increased in a linear fashion. And I just wanted to sort of ask more about that graph. And yeah, so I guess because that study was done in 2008, if there was any more recent work done on sort of, species, is that still the case that it is a linear sort of trend like that? I don't know if that made sense.
Yeah. Jan, can you respond?
Yeah, as far as, can you hear me?
Yes.
Cool. Yeah, as far as I know, it was a linear trend with just the, I think, like smaller amount of densities that I was looking at. But perhaps if it was like a larger density that I was using, maybe things would change.
Mm-hmm.
I think.
And if you were using that setup with your drain pipes, do you think it'd be quite useful for measuring other sort of abiotic variables, like the nutrients in the soils and things like that?
Yeah, I think it could be cool. I think there's some pretty high-tech, larger ones that are used for really looking at nutrients. But I think it could be cool to, like, I could take soil samples and get nutrients that way. Mm-hmm. I think getting nutrients out of the grass is possible as well. Yeah, I think they're pretty cool. They do have limitations though, but yeah, it's pretty good at measuring lots of stuff, I think.
Yeah, it was a really neat study. I really liked the way you could cut the grass quite easily.
It wasn't that fun. There was lots of it.
Yeah, I can relate, but yours seemed a bit cleaner.
Thank you.
Yeah, thank you.
Can I jump in with a question? It's probably more for Tom and Nisa at the start if they were there. But I've just noticed that cattle are now being fed seaweed, and I'm wondering if anyone knows how that affects beetles downstream with dung quality and attraction.
I'm not really sure how to answer that because it's not within my scope. I'm really sorry about that. Yeah. Apparently, Tom was not. He's not here.
Sorry, that's all right. Sort of.
Sorry. Yeah.
Just as a thought, Tom, Dan, just a guess. The seaweed's being fed to cows to stop methane production, and so that's bacteria in the foregut. And it'd be interesting to know how many bacteria in the foregut survive through the hindgut. Like, what's the change in bacteria and archaea along the gut? And so the whatever's living in the hindgut might be responsible for most of those volatiles in the dung. But I think that'd be an interesting thing to look at. I mean, I can't remember the name of the procedure. It's just popped out of my head. it's common in animal science studies when a connection between the stomach and the outside world is placed in the animal surgically.
Yeah.
Fistula. A fistula.
Sorry?
It's a fistula.
Fistula. Thank you, Paul.
Yep.
So
We used to have them
given,
at Cornell.
yeah, so I would imagine CSU have fistulated animals, given that
I
they
haven't
are such
seen
big-
any.
So you might
We
be
do
able
have,
to,
yeah, we do have cattles.
yeah.
Yeah.
So I'm just wondering if you could take a sample of the foregut, the chime out of the foregut. You know, is that attractive to dung beetles? Just as a thought, you know, just a simple
Yeah.
behavioural bioassay.
Thanks, dude. It's a nice idea, actually.
Yeah, and then you could test, and if it is, then you can try with and without seaweed to see what happens.
Yeah, actually, I was able to see lots of differences in these dung profiles with the time and with the temperature, blah, 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 a lot of factors. So it's a microbial activity, of course. I didn't touch that part because that's a lot of work. And then it's like, that's why I didn't have enough time, but I'll have a look if it is something that can be done. Yeah, I'll have a look.
It's one of those things I'm thinking about with your study. So horse dung, hindgut fermenter, more attractive. And farmers often tell us that they get more, they see more dung beetle activity. If they have horses, they might see more activity around their horse yards compared to their cattle yards. And so those anecdotal observations match with your bioassays. So I'm wondering about bacteria and archaea that live in the hindgut compared to elsewhere. Is there something that's particularly attractive that they produce?
Yeah.
But
Interesting.
at the same time, cattle and sheep dung were quite a bit different, and they're both ruminants.
Yeah. Yeah.
Do you think that rearing dung beetles on horse dung would be better?
Yes, that's a great question, Fez. That was my question to Val and Alberto, whether or not they had tried using horse dung in either in Canberra or in Montpellier.
I can answer that question, Theo.
Oh, g'day, Pat.
G'day.
Go for it.
The problem with horse dung is that often, you know, the places you might likely get horse dung from, they're very good at horse owners, and Paul probably might be able to refute me on this or whatever, and I guess it depends on the horse owner, use a lot of agvet. So not necessarily ideal for beetle health, of course, as we know. We went back in the early days in Geelong, we tried using horse dung, and that's what we discovered. We collected it from the nearest horse place, which was the Geelong racecourse, and of course racehorses are drugged up to the eyeballs, so not very successful at all.
On the horse owner, and you know, for example, we'll deworm our horses maybe twice a year, but not very frequently. So you've got lots of time during the year when you would have your dung that would be free of any toxic residues.
I could also add the fact that to collect horse dung presents some practical issues. At least for us, for example, it's much more difficult to find abundant source of horse dung, and so from a, let's say, applied and practical perspective, it becomes a question that would be probably interesting, but even if we have a positive result, then we still would have a big problem because we need to store a lot of dung for the year, and it would be very difficult to obtain that amount.
And that goes the same with sheep dung too. It's a lot harder to collect sheep than it is cow, and you got to weigh up the the benefits and the time and the effort and manpower, women power, all that sort of stuff. So
yeah.
So the cow has always traditionally been the go-to dung, I guess, just for all those other benefits of collecting it. And also, you know, we find certainly because it's involved in food production, you come across more organic growers and all that sort of stuff, which is certainly beneficial for the likes of Alberto and Val and Soletta and I.
So just a thought, something maybe we can play with, but if you want to buy weed seed free sheep manure for your garden, and I have just bought a metric ton of the stuff, because, you know, let's face it, Perth is basically a beach dune, so getting anything to grow here is very hard. It's sourced from animal holding yards, and so they just sweep up the yards. These are the yards on the outskirts of Perth. And so they are feeding the sheep a mixture of pellets and hay. So, and in terms of getting a lot of sheep dung quickly, that might be an option. I mean, there are sheep holding pens like this all around Australia.
I guess it comes down to quality, you know, moisture content and all that sort of stuff. Now, I collected from an organic sheep farm near Canberra just before lockdown, and it was nice and large, juicy, and the beetles just loved it.
Yeah,
it was very, very popular. But trying to get enough of that sort of quality, is very difficult. And just, you know, Tom's talk earlier regarding pasture types and things, like, you know, the pastures here are sort of, you know, alpine, you know, because our organic growers have, you know, they don't improve generally, and they're on natural pasture. You know, we're up against it just to what's available to us.
So Fairs, I think we have to go to New Zealand because they have the sheep and the quality
pasture. Import.
The source of the best sheep dung around.
Please come here.
Send us some as well. We are looking for sheep dung.
Send us some beetles. I actually have a question for Marcella. It's really interesting to see economics coming into these sorts of studies as well. I wanted to ask if you could share a bit more about how the UK entering the EU showed up in your model.
I'm on mute.
Oh no.
Sorry, because we are all in the same room here. Yeah, so you asked about the UK entering the European Union in the same time as the dung beetles introduction. So can you repeat your question, please? Sorry.
So I don't know, I may have taken this note down wrong quickly, but you said that when the UK entered Europe, it showed up in your model, did it?
Oh, yeah, that's
sorry, I might have missed why.
Yeah, no, what I was saying is that we didn't see a significant effect with some of the areas, and that might be because there is some noise, because our analysis starts in 72, which is the year of the first dung beetle introduction, and that was also, we are looking at stocking rate, and that was also the same time as the UK entered the European Union. So that would affect the stocking rate in Australia anyway. So yeah, so it's a little bit to control for that.
So I'll just jump in there if you don't mind me, Marcella. For those of us who were around in Australia in the mid to late 70s, even though I was young, there was a huge impact when the UK joined the EU in Australia and New Zealand as both countries. So both Australia and New Zealand had almost unfettered access. I would not completely unfettered, but there was there was a huge amount of trade from both countries into the UK, and that trade was decimated. So there was across a lot of industries, but obviously a lot of agricultural industries. So I imagine it will have an impact in both countries, a really big one. And businesses in Australia and New Zealand had to go looking for new markets. A lot of farmers started growing different things or changing, sheep from wool to meat or so on. So there's very likely going to be an impact.
And also, I think there is, I think the price in the early 70s of livestock products was quite high. But after that, after the UK entered the European Union, also there was an oversupply of livestock products. So the price in Australia dropped a lot. So a lot of farmers would have, maybe decreased their stocking because prices went down quite a lot. But for that, we can control in our model because, we have price data available. So, yeah. Sorry, I don't know if Val is still here. Yeah.
I am, but I might disappear because there's big thunder and.
Okay. Yeah, I was just wondering with the emergence rate or success rate, if the brood ball size or the egg size had any, if was very different between like species and things like that.
Between species, the two species we mainly worked on have same size eggs. So
Right.
Ontophagus vacca and Ontophagus andalusicus. So one of them doesn't do as well, but I don't think it has to do with anything like that. They're just a bit less productive. Sturmey, the one that Alberto talked about, is a much bigger egg because it's a bigger beetle, bigger egg. And actually the process we have to do, the surface sterilization needs to be tweaked a little bit because they are more fragile as well. So that's a bit bigger egg, more fragile. And so the process is actually a little bit more difficult. But for the, so it's not really, I can't really tell if with the bigger egg, if there's better emergence rate. It kind of varies from box to box. Right. Sorry, it's not very a useful answer. Yeah.
It's a Val. Can I ask a question? Something I wanted to ask about at the end of your talk, but I love the idea of the magic mix.
It is magic.
So my question is, let's say for Sturmey, just as a beetle that's going to come into Australia, where's the magic mix going to come from? Is it going to come from other species that are already in Australia? So you choose female beetles, I don't know, Ontophagus taurus or whatever it might be, and you use their gut extracts, or are you planning on getting Sturmey mothers in Morocco and somehow I'm confused.
No, we can't do that. It's actually quite tricky, because it's species specific. So we've started playing with that, and so we used, because we're now processing Andalusicus. So I talked about vacca, but we have Andalusicus now that are coming out of quarantine, and because we have vacca that are outside quarantine that have gone through several generations, we decided to use those females because we figure out they should have enough microbiome by now that we can use for our other species, for Andalusicus. So our magic mix ended up being not magic. So because it was a cross-species thing, it didn't actually work really well. And I haven't looked at the data closely, but it doesn't seem to have worked. So it is kind of species specific. So what the next step, I think, is to figure out what's functional in the microbiome, and then see if we can actually make a microbiome with those elements that are important.
Yeah.
But that will be, that probably won't be part of this program because it's quite a big chunk of research. But I think that's what we have to aim for. Because if we can actually make a microbiome with the functional elements, then we can just inoculate our beetles the eggs with that. Yeah, but they will pick up microbiome from, they will pick up microbiome from the environment as well. It's just, it takes longer and they're not very fit compared to if they had their maternal microbiome. So
Val, do you think the essential components of the magic mix are organic compounds or living things, microbes?
I reckon they're microbes. They should be microbes. Yeah.
So I think that can be really tricky.
Yeah, we can't take them out of quarantine. That's the thing. You can't, you can't take out anything from quarantine. So we have to find a way of making a microbiome or finding a surrogate species or something like that we can use for dung beetles. But so far we haven't found anything that works. But we've proven that we can inoculate, just like one step.
So are you going to use 16S or whatever marker on beetles in Morocco? So say Andalusicus, for example.
Yeah.
Look at the original population in Morocco and or France or Spain. What's in their microbiome compared to the ones that are here. So I'm just trying to just to see whether or not those microbes can be picked up in Australia. Are they even present here? Are they sort of moving in our environment? And we've got 8 minutes, by the way, people.
Okay. Well, it's something we could do, but so from this experiment that we've done, we will look at everything and we'll see what's in common, and so we and then we can at least see what's kind of missing from our control at least, and what's actually in the magic mix to make these big beetles. So if there's more of something, maybe we can kind of play with that and try to isolate it or do something. But yeah, ideally it would be if you think about future research, I think that's what you would do. You would look at what is in native beetles, see if we can find the same thing that is functional, and then just use those from the outside world, not from what we bring from quarantine. So there's a lot of different questions we can ask.
Yeah. So has anybody looked at the microbial fauna in the maternal gift or the pedestal of these egg masses of any species of dung?
Yes, but not off the top of my head. So there's quite a bit of research that is done in a group in the US. We're actually collaborating with Armin Matzek. Yeah, I think they have done it, but not from the top of my head. Yeah, they definitely have proven for Ontophagus anyway that actually the maternal gift is what is really important for the fitness of the beetles.
Yeah, there's so many. It's been a great symposium. Lots of lovely new information and data coming out. But like with everything, more questions. Well, we've got about 5 minutes before this Zoom link becomes the Zoom link for everybody. So I think we might just wrap up. And many thanks to our presenters, Tom, Nissa, Marcella, Dan, Fez, Alberto, Val, Megan, and Paul. Thank you very much. That was fantastic, terrific talks. And I'm sad that we weren't all able to meet up in person, which was our original hope, but we live in interesting times. So I'm hoping the times get less interesting. And certainly we in the West would love to come and visit people over East and see you face to face for a change, hopefully next year, and for another meeting. So thank you very much, everybody, again, and for an excellent set of presentations. and have a Merry Christmas and Happy New Year. And we will see you again very soon.
Okay, thanks all.